So our webinar today is a new perspective on relationship between brain and behavior. I'm really pleased to have Henry Yin. Henry received his Ph.D. in psychology from UCLA, worked with Barbara Knowlton and Bernard Boline on the role of basal ganglia in learning and habit formation. Did his postdoctoral fellowship in David Lovinger's lab at NIH, where he studied the mechanisms underlying striatal synaptic transmission and plasticity. He's currently Associate Professor of Psychology and Neuroscience at Duke University and a faculty member of the Neurobiology Graduate Training Program and a member of the Duke Institute for Brain Sciences. And among his many publications are a few that our office has supported either through the OPNet Program for Basic Behavioral and Social Sciences Research, or co-funding along with some of our many IC colleagues that have neuroscience interests in their institute. So with that, Henry, I will turn it over to you, and thank you again for joining us today.
Okay, thank you, Bill, for that kind introduction. So in today's talk, I will first review some recent studies from my lab on the relationship between neural activity and continuous behavior. Then I will introduce a hierarchical control model that can explain these findings. And finally, I will discuss some implications of this model. Now, the traditional paradigm in neuroscience simply assumes that the brain is some sort of input-output device. The typical approach, as you can see here, is to vary input and measure behavioral output. For example, here you have a head-fixed monkey sitting in a chair, and this is after many months of training to get him to sit there, and then a sequence of stimuli is presented. So you first have a cue, some target, and then some delay, and finally he gets to do something. So in this paradigm, behavior is really reduced to a sequence of discrete events. And this is convenient because it can be recorded simply as a sequence of timestamps, and neural activity can be measured in relation to these timestamps. So I will call this the event-based approach. It's simply assumed that nothing is really happening between events. So basically, between events, the brain is somehow turned off, or whatever it's doing is not important. Now, in my lab, we take a different approach. I like to call it the process-based approach. And that means that we treat behavior as a continuous process. So here's an illustration of video-based motion tracking. Here's an animal, a mouse, sitting on a platform with a wireless test stage. So we can also use 3D motion capture to quantify behavior. We can put reflective markers on the body of the animal, and then use cameras to capture motion continuously. So this is what the data might look like. So essentially, this is a mouse moving around. So if we combine this with wireless recording of in vivo neural activity, then we simply end up with behavioral measures like position, velocity, and neural measures like firing rates and different types of neurons. So all this seems simple enough, but I think this approach is actually new, and as we shall see, it has produced very surprising results. So first, I will describe some of our results using this approach on the basal ganglia output from the substantia nigra pars reticulata. So here is the SNR, pars reticulata, is the major output nucleus of the basal ganglia circuit in mice. And it receives direct projections from the striatum and in turn projects to areas like the midbrain, brainstem, and thalamus. So in this study, we simply measured, using a video camera, measure the, in 2D, the position of the head of the animal, which is, captured by this LED on the head stage of the animal. And the task is very simple. There's a sucrose delivery spout close to the animal, so the mouse simply has to get the sucrose whenever it's delivered. Okay, and then we can measure X and Y position of the animal. Now here's an example of representative data from our wireless recordings. What we have here is a raster plot of the individual spikes. Okay, so in black you can see individual action potentials, and so each row is a single trial. And the basic finding is that if you look at the neural activity, action potential, firing rate, and the X position as captured by the video, there is a remarkable relationship. So that for each individual movement, there's a high correlation between the spiking of basal ganglia output neurons and X position measures. Okay. So if you look at this plot here, you can see that here's the Y position in green, X is in blue, and the neural activity is in red. Okay, so they're remarkably similar. So in other words, we have a neuron that's whose firing rate is correlated with the X component of the position vector. Now, to make a long story short, we found that the basal ganglia output neurons represent the instantaneous position coordinates of the head. And with 2D video tracking, we found 4 types of neurons corresponding to the 4 directions, up, down, left, and right. For each axis of motion, there are opponent neural outputs. So, for example, up and down, they are opponent. Left and right are opponent. And so in normal movements, a combination of these will be activated and suppressed accordingly. Okay. And so basically for each axis of motion, for example, horizontal motion or vertical motion, we have a push-pull system. For horizontal movements along the x-axis, you can have 2 directions, left and right. Okay, the neutral position or the origin of the egocentric reference frame here is somewhere in the middle. So that's close to the middle of the dynamic range of this neuron. Now, if you move to the right, the opponent neural outputs will change their firing. So there are 2 types of neurons will change their firing rates accordingly. And if you move in the other direction, you get opposite changes in firing rates. In the middle, so remember that this output neuron can be tonically active. So it's actually firing at, let's say, 10 hertz or 20 hertz. So these output neurons are basically pacemakers, but they can modulate their firing rates based on the direction of movement. So these are just arbitrary numbers. Basically, you have opponent outputs. If you're moving in one direction, this neuron is increasing their firing rate, its firing rate. And this is the middle, this is the neutral position. And for the opposite type, it's decreasing as you move in this direction, but increasing in the opposite direction. And of course, the sum will always be the same, and the difference would be the actual signals that are sent, I think, downstream of the basal ganglia. So that means that when we just take a pair of these output neurons, let's say X and Y neurons, we can reconstruct the body position vector at any time. So the basal ganglia output at any time will give us the instantaneous position of the animal. And it also follows that the rate of change in the basal ganglia output reflects movement velocity. Now, our results actually contradict the textbook model of basal ganglia function. So if you ever took a neuroscience class or went to medical school, you'd be familiar with this model. What it says basically is that you have these tonically active GABAergic neurons that suppress behavior. And when you have a pause in the output, that essentially opens the gate. Okay, it allows action selection. The problem is that the action in this sense is event-based. So it's really a discrete event. It's all or none. Okay, but our data show that this assumption is wrong. Obviously, actions would involve changes in body position, but that's a continuous process. And if you measure behavior continuously, you can see that it's it's not discrete. So here's the traditional model, which is like a excuse me, on-off switch, and so these are the well-known models, essentially selecting the desired action event and suppressing all other actions. But what we have shown is that it's better to describe these outputs as continuous dials. For example, we have X and Y independent systems. And so you can modulate firing continuously throughout voluntary behavior. So use another metaphor. The traditional metaphor is simply that the basal ganglia is some sort of gate that's occasionally opened briefly and sort of allow behavior to run out, if you will. And but I think a better metaphor is that of a conductor who's continuously sending information to many musicians, telling them not only whether, but also how quickly and how much. Okay. So the critical question still is exactly what does the basal ganglia output do? To answer this question, we have to look at the major output, the major targets of these outputs. So as I mentioned before, they target the thalamus, midbrain superior colliculus, and pedunculopontine nucleus in the brainstem. And the first thing to notice is that these outputs do not activate muscles directly. So we actually know something about the target regions. In the 1940s, Hess did some classic experiments in cats using chronically implanted electrodes to stimulate various parts of the midbrain and thalamus. And he found that stimulation in different regions produced distinct movement patterns. In particular, he found cell groups that, when stimulated, produced either horizontal or rotational or vertical movements. So there's a map of different classes of movements. And what's surprising is that these are relatively pure movements, which indicate that there are independent systems in these target regions that control different axes of motion. So when you generate a volitional movement, the basal ganglia output can recruit the right combination of these systems. Okay, so there appears to be independent downstream systems for at least in horizontal, vertical, and rotational movements. Obviously, we only identified the horizontal and vertical components, but I wouldn't be surprised if we find rotational movements, cells related to rotational movements in the future. Now, the other important question is, why is the basal ganglia output related to position? Right? So, as I mentioned, it does not activate muscles directly. In fact, if you look at the neuroanatomy, what happens is that it's mainly projecting to the reticulospinal pathway, and then in turn to the spinal motor neurons. So it seems that these descending signals are in a position to influence the muscles indirectly. So it would make sense that these are actually related to proprioceptive perceptual inputs related, reporting position. But why are these output signals related to actual position achieved? Now, I think to answer this question, we first have to consider the ultimate output of the nervous system. Okay, so as we know, for all behaviors, you actually have to activate the motor neurons. Okay, so the relevant output is from the alpha motor neurons to muscle fibers. And so the muscles are the major effectors. Sherrington called this the final common path. All behaviors are produced by this output, but there are multiple descending pathways from the brain that can influence the final common path. However, although the final common path would produce some sort of force output, some increase in muscle tension, it would be a mistake to equate the neural output from the motor neurons with behavior. And that's because the output of the motor neurons can only generate some force, okay? The actual achieved position will depend not only on that force, but also environmental disturbances. So in 1935, Bernstein wrote that actually there are no situations in which muscle shortening is the cause of a movement. And this is a very deep insight because as Bernstein realized, if you repeat outputs from the final common path, you might repeat force outputs roughly, but these cannot achieve repeated positions because the loads will change depending on various disturbances like gravity, wind, and fatigue, and so on. And these disturbances can alter the actual consequence of muscle contraction. So this fact has profound implications, I think, for understanding the relationship between behavior and brain output, because the observed behavior is actually not just output of the muscles. Okay? It's not the product of the muscle output alone, but of both muscle output and some other force from the environment. And so when we look at the neural output, that's only half of the story. So there are unexpected disturbances, and which basically makes it impossible to repeat behavior by repeating muscle output. So the question is, how do we do it, right? So I think there is a tremendous computational challenge because if the output from the motor neurons does not determine the observed behavior, then how can the brain produce just the right amount of variations to counteract the effects of environmental disturbance? Traditionally, to solve this kind of problem, it's assumed that you simply have to anticipate all future disturbances and you calculate you have some sort of internal model of kinematics and dynamics. So to make a long story short, I think this approach has failed. And the reason it failed is that it basically assumes that all animals that move can understand physical laws perfectly and compute these laws in real time and to use effectors that never change. I think that really vastly underestimated the complexity of the actual environment and overestimated the capability of the nervous system. So what's the alternative? Well, I think there is a simple alternative, which is simply negative feedback control, which unfortunately has been misunderstood or ignored for decades. So in a control system, what's happening is that there's an input variable derived from the sensors, and that is the signal is being sent to a comparison function, which is comparing some internal reference signal and sensory input. And the discrepancy or the difference or the error signal is used to produce the output. So the internal reference signals represent desired states, and the trick is to specify what you want, and the output will automatically vary accordingly without doing any computations, because it's actually embodying the computations. All you have to do really is subtraction. So I think the negative feedback control system is the basic unit of control, and in the nervous system there are many control systems like this organized in a hierarchy. And the way this works is that the higher levels can send reference signals to lower levels. So these are sort of like commands that can tell or request the lower systems how much input to sense. Okay. So here's an illustration of how the brain or how the nervous system can implement the control hierarchy. And as you can see here, the cortico-basal ganglia system, which I talked about, is really situated at the highest levels of the hierarchy, okay, above the lower systems would control orientation, body configuration, posture, muscle length, and muscle tension, but this is the highest level. So if you request a specific position to be achieved, for example, when you're standing or sitting, then the negative feedback organization will actually achieve that position. But when you do that, all the lower level systems must vary their reference signals and their outputs as well. So it's actually a very complex hierarchy. Now, earlier I talked about the basal ganglia output. So obviously, one question is, what about the input, this striatum, which is the major input to the substantia nigra pars reticulata? And so the output, as I said, is related to position, but the input, actually the input nucleus activity in the striatum is related to velocity. And so this is an optogenetic experiment, simply stimulating, so these are light pulses, stimulating the striatal neurons, and basically the frequency of stimulation determines the speed of movement. Okay. And if you record from the striatal neurons, this is a medium spiny projection neuron from the striatum, and these are this is a raster plot of a medium spiny neuron, so firing rates, and here you have speed of the animal, rightward movement. And as you can see, there's a linear relationship between the neural activity and velocity. And same is true if you look at the nigrostriatal dopamine neurons. So these are the neurons that modulate striatal activity. They come from the substantia nigra pars compacta, and they project to the striatum. And these neurons are also related to velocity, very similar to the striatal projection neurons. And as you know, in Parkinson's disease, what happens is that these dopamine neurons die, and consequently you have slowed movements. Okay, so when you have a disruption in this velocity control system, you have obvious symptoms, most prominently inability to initiate actions and slowness in movements. Now, so we have proposed that this is really the fact that the striatum contains velocity-related neurons and the substantia nigra pars reticulata contains position-related neurons. This is a very important clue because it tells us that there is a hierarchical relationship. So basically what we have here is a velocity loop above a position loop. And the basal ganglia circuit is doing velocity control. And in the output function of the circuit, there is a leaky integrator that's integrating the velocity signal continuously and produces the position reference signal. So this is telling the downstream position controllers. So remember, you have the independent controllers according to the classic results from Hess. And the magnitude of the velocity error signal is proportional to the rate of change in the integrator output. So that's by definition what the integrator does. So in other words, the basal ganglia output, according to this model, is a position reference signal to downstream target areas which contain position controllers. Now, obviously, So far, I've talked about velocity control, which is really a type of transition control. In velocity control, you just control the rate of change in proprioceptive input. That's what you find in the car, but clearly the function of the basal ganglia is not limited to velocity control. We know there are these parallel basal ganglia networks, and so obviously one question is, what about the other networks, right? So one possibility suggested by the anatomy is that these other basal ganglia networks can be used to control different types of transitions. And what all these transitions have in common is the rate of change in some perceptual variable. So the rate of change itself must be explicitly represented and controlled by varying outputs. So that means that continuous measures are absolutely critical. So our model raises an important question, which is what is above the basal ganglia? How do the higher levels command the transition controllers? So if the basal ganglia networks contain velocity controllers like a car, then who's the driver, okay? And I think it's important to appreciate that the same action, like raising your hand, can be used for any number of purposes. So you can raise your hand to vote, or to indicate disagreement, or to get food. The reason for the action is actually hierarchically higher. So we know there are massive cortical striatal projections from the cerebral cortex to the striatum. And so far I've talked about the output of the basal ganglia, and what about the input to the striatum, right? So the striatal neurons, which reflect velocity, must be commanded by some sort of input. And the question is, what do these signals represent? Now, obviously, there are many possibilities, as many goals of behavior can be abstract and difficult to measure. So the question is, which variable must be controlled for most voluntary behavior? And I would argue that a common variable is proximity to target. So as you can see here, in most voluntary behaviors, there is some sort of spatial target. And it could be stationary, could be moving, but one variable that we have to control in order to, for example, reach for a cup or hit a tennis ball, is to control that distance between self and target, okay, some part of the environment. And we designed a task to do that, to study target proximity control in mice. So we simply have a moving target with a sucrose spout. So the sucrose is being delivered continuously, and the target is moving basically horizontally in the x-axis, okay? And we can measure the target position with a marker and the head position of the animal with another marker. And we can then record from the striatum, in particular, this circuit that receives corticostriatal input, the spinal projection neurons, which the medium spiny projection neurons, which I mentioned earlier, and also the fast spiking interneurons. So these neurons receive cortical input, and the question is, during this sort of pursuit behavior, what happens to the firing rates of these neurons? And as I mentioned earlier, the striatal projection neurons, the medium spiny neurons, just represent velocity. So here's one example. Here's a velocity measure from, 0 is leftward movement. And so here's the velocity measure. Here's the neural activity, okay? A very high correlation. Here's rightward movement, and so now the firing rate is reduced, okay? But the correlation is also very high. But more interestingly, in the fast-spiking interneurons in the striatum, we actually found that there is a representation of the proximity to target. Okay, so the distance between head and target is represented. So again, so here's the proximity changes over time, and here's neuroactivity from the fast-spiking interneurons. Again, the correlation is very high. And if you simply if you inhibit the activity of these neurons with halorhodopsin optogenetically, you can disrupt the pursuit behavior. So you actually don't affect behavior, or you don't reduce velocity in general, you don't affect movement, but you affect the accuracy of pursuit. Now the animal can no longer follow the moving target accurately. So here is a measure of the error, pursuit error, and this measure is much higher when you inhibit the fast-spiking interneurons. So the basal ganglia input from the corticostriatal projections then can, I think, command velocity controllers downstream, and the higher order representations such as distance to target, which is the pursuit error, are used as descending reference signals, as suggested by these results. Now, normally when I give a talk, I would stop at this point, but I think there are some key implications which I'd like to discuss briefly. I think the results I just described are actually surprising if you view them from the perspective of traditional neuroscientists. They tell us something new about the nature of neural signals and the relationship between brain and behavior, and finally how to study behavior. So the conventional wisdom is that there's a complex relationship between brain activity and behavior, but we actually showed that there's a linear relationship, and I think it's because there has been no serious attempt to measure behavior continuously. So is it really that simple? I think it is. It's actually that simple. I think this is why it's extremely important to have continuous measures. So when we measure the behavior carefully and continuously, the relationship is surprisingly simple. It's linear. Whether we're talking about position or velocity or distance to goal. And the neural signals are also one-dimensional. So traditionally, the assumption is that you have some sort of digital transmission. So that's based on the fact that the action potential is all or none. And so each spike is a digital or logical variable. And we've shown instead that really this is a continuous and analog signal. So we're not talking about the subthreshold membrane potential fluctuations. I'm actually talking about the firing rate itself. So for the peripheral receptors, for example, in the classic work from Adrian and Mountcastle, people have found the linear relationship and the analog nature of neural signals. But in the brain, as far as I know, this is really the first demonstration of widespread analog signals representing discrete physical variables. And in other words, I think the traditional assumption that you have encoding and decoding of information is actually misleading. In fact, what we're dealing with is an analog computing device using instead of using symbols, you're representing physical quantities with another physical quantity. And in electronic analog computers, the quantity is simply voltage. In the brain is firing rate. Now, another implication of our work is that, so remember that for our behavioral measures, we're recording for the 3D motion capture about 100 frames per second, and that's close to 1 million data points in a few hours, okay? And the interesting fact is that I think it's better to sample behavior in one individual this way, to acquire 1 million data points from a single animal, than to acquire 1 data point per animal and use 1 million animals. And because we can see the continuous relationship between the neuroactivity and behavior in the individual animal at any time, okay? So it really doesn't make any sense to average data from multiple animals using this approach. If we did that, we would miss the crucial relationship, the relationship between neuroactivity and the control variable, and that's the relationship that actually explains how behavior is generated at any time. And so I think the main problem is that, as I mentioned before, the traditional assumption is that you simply measure output as a function of input. And there's some sort of sensory motor transformation, and when this doesn't work, it gets very noisy. Then you would use very fancy mathematical transformations to decode the neuroactivity. And what we found is that this is unnecessary. In any case, you can't even do that with a closed-loop system because the input is affecting output at the same time that the output is affecting input. So the output is really not a function of the input, but when you look at the actual control variable, the relationship between the reference signal and the achieved sensory inputs is highly is basically highly correlated. It's essentially the same because you have control. Okay, so behavior is not output, but a consequence of controlling some input. Okay, so I like to emphasize the circular causation in the closed-loop system. And finally, I think traditional studies focuses on the average animal because what you're looking at is some sort of statistical trend, okay? And the conclusion does not necessarily apply to each individual in the group. And our studies show that, on the other hand, we can actually understand what the individual is doing at any time because we have a working model that makes quantitative predictions. Of course, so far we have only studied the higher level variables. We have only studied the variables that we can measure from the basal ganglia. But the model of behavior that I just described is actually a working model. That means it's the kind of model that can be built eventually, and I think we will. So I'd like to end with a very important point, which is that the study of behavior, in my opinion, is an exact science. It's not statistical in nature. And I think our work suggests that we can actually understand the individual because the control process is a continuous process that can be only understood fully if we identify the components of the individual control system and the level of the control hierarchy within each organism. So I hope I can persuade at least some of you that it's time to restore the idea of the individual in studying behavior. Okay? Now, in the future, I hope we can talk about brain or control hierarchy as a singular, so singular individual brains, not average brains, and also we have to take into account the fact that to understand any behavior, we have to identify the control variable of the individual at a specific time. Okay, so finally, I'd just like to acknowledge my lab members, especially Joe Botter and Namsu Kim, who did most of the work I talked about, and my funding sources, and obviously OpNet and OBSSR for support. And thank you for your attention.
Great, Henry, thank you. That was great, and particularly like the focus on more computational modeling, some of these closed circuit modeling approaches, as well as sort of the reminder that we can learn an awful lot from doing intensive longitudinal within subject studies as opposed to between subject comparisons. I'm going to turn this over to Bill Elwood to moderate the Q&A. Bill coordinates our webinar series, and then those of you who are on the phone, if you'll type in your questions, we'll get those started. So, Bill, I think you had actually sketched out something as well as a question, maybe?
I did. Thank you so much, Henry. Your point about a million data points on one person versus, one data point from a million people resonates with NIH's current work on reproducibility. Do you have any comments about how your work fits in that larger scientific discussion?
So for example, we've done this with many animals, and obviously we have always been able to replicate the results. So in that sense, it's almost trivial because we always find the same thing when we try to replicate. But I think the key is that if you're not getting quality data from an individual animal, and then if you use a lot of animals, you might see some statistical trend, but it's hard often to interpret what that means. And traditionally, a lot of the interpretations are problematic because you don't know exactly which variable is appropriate. And I think identifying that variable for the individual animal first is the most important step. And
actually very similar to a lot of the thinking in single case studies or single control studies, where people basically say, each new individual that you evaluate intensively over time becomes your replication of the previous one, and actually some nice Bayesian modeling on how you can extrapolate from that to predicting what the next individual would probably do under the same circumstances. Let me ask you just a quick question as well about developmental processes. Your information about the self to target, I was thinking about in infancy that's not a well-formed phenomenon, right? The ability to be able to know where you are self in space and also be able to, in reference to a target. Has any of this work been done developmentally to determine how that gets developed and built and what the behavioral exposures are that cause that to happen?
yeah, I think that's a really interesting question. No, I don't think anyone has done work on that. In fact, we haven't even looked at how this develops in the mouse, because obviously there's a learning process. So to pursue a moving target accurately, even the mouse has to learn for a few hours. In the beginning, it's not going to be as good, right? So there's a learning process involved, and I assume there's definitely plasticity in the corticostriatal system, but I just don't know, exactly what's changing. And if you have to study this in young animals, baby mice, obviously the real technical challenge is simply measuring the behavior. So you can imagine it would be very interesting to study this in young animals, but it would also be difficult to track the behavior and have a relevant target that it's actually interested in. So I think developmentally there's a lot of potential, but there are some practical problems that we have to solve first.
Exactly. That makes perfect sense. Well, given time, I wanted to thank you again and thank all the attendees on the webinar for joining us. Apologies for some of their audio difficulties, and we'll keep working on our systems until we get them right. But Henry, I want to thank you again for taking the time to do this today.
Okay, thanks.
All right, thank you. All
right. Bye-bye.
Bye-bye.